Chapter 8, Cell, the Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. 8.1 What is a cell? Unicellular organisms are capable of first independent existence and second performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton van Leeuwenhoek first saw and described a living cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. 8.2 Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Schwann, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Schwann proposed a hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omnicellulae e-cellulae. He modified the hypothesis of pre-existing cells, unicellular e-cellula, who modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as understood today is: first, all living organisms are composed of cells and products of cells. Second, all cells arise from pre-existing cells. 8.3 An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall and as its outer boundary, and just within it is the cell membrane. The cells of the human cheek and just within and have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called a nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material DNA. Cells that have Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called the cytoplasm occupies the volume of the cell. The cytoplasm is the main area of the cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound or distinct structure called organelles like the endoplasmic reticulum (ER), the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cell lacks such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and or rough ER. Animal cells contain another non-membrane-bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, activities. Figure 8.1. Diagram showing different types of shapes of cells. Red blood cells, round and concave. White blood cells, amoeboid. Columnar epithelial cells, long and narrow. Nerve cell, branched and long. Tracheid, elongated. Mesophyll cells, round and oval. For example, mycoplasmas, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich among multicellular organisms. Human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function of they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, fluoropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. Figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria: Bacillus rod-like, coccus, spherical, Vibrio, comma-shaped, and Spirillum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotic all prokaryotes have a cell wall surrounding the cell membrane except in Mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, a single chromosome or circular DNA, many bacteria have a small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. Figure 8.2 diagram showing comparison of eukaryotic cell with other organisms. A typical eukaryotic cell, that is 10 to 20 micrometer. Typical bacteria 1 to 2 micrometer. PPLO about 0.1 micrometer. Viruses 0.02 to 0.2 micrometer. 8.4.1 Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, which is those that take up the Gram stain are Gram positive, and others that do not are called Gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain the pigments. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made up of special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S subunits, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called a polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins, inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm, for example, phosphate granules, cyanophecin granules, and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes include all the protist plant, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into the chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have a centrioles which are absent in almost all plant cells. Figure 8.3. Diagram showing plant cell. Rough endoplasmic reticulum, nucleus, nucleolus, Golgi apparatus, nuclear envelope, plasma membrane, vacuole, middle lamella, cell wall, mitochondria, ribosomes, chloroplast, cytoplasm, peroxisome, microtubule, plasmodesmata, smooth endoplasmic reticulum, lysosome. B. Animal cell, microvilli, plasma membrane, centriole, peroxisome, lysosome, ribosomes, mitochondria, rough endoplasmic reticulum, cytoplasm, nucleus, nucleolus, nuclear envelope, smooth endoplasmic reticulum. Let us now look at individual cell organelles to understand their structures and functions. 8.5.1 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in the bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. Figure 8.4. Cholesterol, sugar, peripheral protein, integral protein, phospholipid bilayer. In addition to phospholipids, membrane also contain cholesterol. The lipid the lipid component of the membrane mainly consists of phosphoglycerides. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the cell membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. Figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the points of view of junction like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport, for example Na plus or K plus pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides a barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactans, and minerals like calcium carbonate, while in other plants it consists of cellulose, semicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards the membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata which connects the cytoplasm of neighboring cells. 8.5.3 Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum ER, Golgi complex, lysosomes, and vacuoles. Since the function of the mitochondria, chloroplast, and peroxisomes are not coordinated within with the above components, these are not considered as part of endomembrane system. 8.5.3.1 The endoplasmic reticulum ER. Electron microscope studies of eukaryotic cells reveal the presence of a network of or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum ER. Hence, ER divides the intracellular space into two distinct compartments, that is luminal inside ER and extraluminal cytoplasm compartments. The ER often shows ribosomes attached to their surface. The endoplasmic reticulum bearing ribosomes at the surface is called rough endoplasmic reticulum. In the absence of ribosomes, they appear smooth and are called a smooth endoplasmic reticulum SER. RER, that is rough endoplasmic reticulum, is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid like steroidal hormones are synthesized in SER. Endoplasmic reticulum, nucleus, nuclear pore, rough endoplasmic reticulum, ribosome, smooth endoplasmic reticulum. 8.5.3.2 Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts or the forming phase and concave trans or the maturing phase. The cyst and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the endoplasmic reticulum fuse with the cyst phase of the Golgi complex and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of trans important site of formation of glycoproteins and glycolipids. Figure 8.6 Golgi apparatus. Its sister name. 8.5.3.3 Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes: hydrolases, lipases, proteases, carbo, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. 8.5.3.4 Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called a tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against a concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for extraction in many cells, as in protists. Food vacuoles are formed by engulfing the food particles. 8.5.4 Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size, also considerable degree of variability is observed. Typically, it is the sausage-shaped or cylindrical, having diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called cristae, singular crista, towards the matrix. Figure 8.7. Structure of mitochondrion: longitudinal section with outer membrane, inner membrane, matrix, crista, intermembrane space. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses singular circular DNA molecule, a few RNA molecules, ribosomes, 70S type, and the components required for the synthesis of proteins. Mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for the trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotenes, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. Amyloplasts store carbohydrates, starch, for example potato. Elioplasts store oils and fats, whereas the aleuroplasts store proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas. A green algae to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. The the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Figure 8.8. Thylakoids are arranged in stacks like the pile of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. The chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller 70S type than the cytoplasmic ribosomes 80S type. Figure 8.8 sectional view of chloroplast with outer membrane, inner membrane, granum, thylakoid, stroma lamellae, stroma. 8.5.6 Ribosomes. Ribosomes are the granular structures first observed in under the electron microscope as dense particles by George Palade in 1953. They are composed of ribonucleic acid (RNA) and proteins and are not surrounded by any membrane. Eukaryotic ribosomes are 80S Svedberg units, while the prokaryotic ribosomes are the 70S type. Each ribosome has two subunits, larger and smaller subunits. Figure 8.9. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S stands for Svedberg's unit for the sedimentation coefficient. It indirectly is the measure of density and size. Both 70S and 80S type ribosomes are composed of two subunits. Figure 8.9 ribosome with larger subunit and the smaller subunit. 8.5.7 cytoskeleton. An elaborated network of filamentous proteinaceous structures present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and flagella. Cilia, singular, cilium, and flagella, singular, flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of a cilium or the flagellum showed that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9+2 array, figure 8.10. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole like a structure called the basal bodies. Figure 8.10. Section of cilia, flagellum showing different parts. A. Electron micrograph. B. Diagrammatic representation of internal structure with plasma membrane, peripheral microtubules, doublets, inter-doublet bridge, central microtubule, radial spoke, central sheath. 8.5.9 Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. 8.5.10 Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interface in nucleus of cell when it is not dividing has highly extended and elaborated nucleoprotein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nucleoli, singular nucleolus, figure 8.11. Structure of nucleus with nucleoplasm, nucleolus, nuclear pore, nuclear membrane. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometers called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places that the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of the organisms that have more than one nucleus per cell? Some mature cells even lack a nucleus. For example, erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called the chromatin, but during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2 meter long thread of DNA contributed among its 46 to 23 pairs of chromosomes. We will study the details of DNA packaging in the form of chromosome in class 12. Every chromosome visible only in dividing cells essentially has a primary construction or the centromere, on the sides of which disc-shaped structures called kinetochores are present. Figure 8.12, chromosome with kinetochore. Centromere holds two chromatids of a chromosome based on the position of the centromere. The chromosomes can be classified into four types. Figure 8.13, a metacentric chromosome as middle centromere, forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called the satellite. Figure 8.13, types of chromosome based on the position of the centromere. Satellite, secondary construction, centromere. Short arm, centromere, long arm, short arm. 8.5.11, microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Summary: all organisms are made of cells or aggregates of cells. Cells vary in their shape, size, and activities or functions based on the presence or absence of a membrane-bound nucleus and other organelles. Cells and hence organisms can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates the transport of several molecules. The endomembrane system includes Golgi complex, lysosomes, and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles but also play a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. ER helps in the transport of substances, synthesis of proteins, lipoproteins, and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The, the secretions of cells are packed in them and transported from the cell. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with endoplasmic reticulum. Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. They are bound by double membrane. The outer membrane is smooth and inner one folds into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions and the stroma of dark reactions. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastids are chromoplasts which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is the structural and functional unit of life.